Hello everyone, this is our fourth chapter, cell division. So explain cell division and life cycle of a cell, meiosis and mitosis with diagrams. So cell division is the most important process for creating new life. As the name suggests, the cell undergoes division to form two new cells, which in turn further undergo division. This is how living organisms are created. Hence, cell division is also called cell reproduction. There are two types of cell division, mitosis and meiosis. Mitosis is found in the somatic cell and meiosis is in generative cell. During mitosis, the cell divides into genetically A-like daughter cells, while in meiosis, two germ cells mix together to form four genetically different daughter cells. In this lecture, we will learn about both these processes in detail with their stages and diagrams. Cell division. It is a process by which a parent cell divides into two or more daughter cells. It is a very important process in all living organisms. All these processes like cell division, cell growth and DNA replication hence have to take place in a coordinated way. The sequence of events by which a cell duplicates its genome, synthesizes the other constituents of the cell and eventually divides into two daughter cells is known as the cell cycle. This is the structure of cell cycle. 24-hour cell cycle, G1 phase, S phase, G2 phase and M phase. In the M phase, this is only for around 1 hour. So all stages are coming in this M phase, means mitotic phase. Other is called resting phase. So now this is interphase. In G1 phase corresponds to the interval between mitosis and initiation of DNA replication. In S or synthetic phase marks the period during which DNA synthesis or replication takes place. In G2 phase marks the period in which proteins are synthesized in preparation for mitosis while cell growth continues. So cells present at the G0 phase to enter in the G1 phase need to enter in a specialized gate present in the G1 phase to act as restriction entry. The cell that do not divide later exit the G1 phase to enter an inactive stage termed as quiescent stage of the cell cycle. During the S phase, the amount of DNA per cell doubles, but the chromosome remain the same. M phase, yani mitotic phase, the number of chromosome in the parent and progeny cells is the same. It is also known as equational division. Mitosis has been divided into 4 stages of nuclear division. So you have to remember, mitosis is called equational division and meiosis is called reductional division. So prophase, this is the first phase. Prophase, which is the first stage of mitosis, follows the S and G2 phases of interphase. The following characteristic events takes place in prophase. Chromosomal material condenses to form compact mitotic chromosomes. Chromosomes composed of two chromatids attached together at the centromere. Initiation of the assembly of the mitotic spindle, the microtubules, the proteinaceous components of the cell, cytoplasm help in the process. Cells at the end of prophase do not show Golgi complexes, endoplasmic reticulum, nucleolus and the nuclear envelope. Second is metaphase. It starts the complete disintegration of the nuclear envelope. Hence, the chromosomes are spread through the cytoplasm of the cell. Condensation of chromosomal materials into compact and distinct chromosomes made up of two sister chromatids attached with spindle fibers with kinetochores. Chromosomes arrange at the center of the cell called the metaphase plate. Anaphase, splitting of each chromosome at centromere into two sister chromatids. Two chromatids start moving towards opposite poles. Last one is telophase. Telophase is the last stage of mitosis, which involves chromosome cluster at opposite spindle poles, and their identity is lost as discrete elements. The nuclear membrane reassembles around the chromosome clusters. Nucleolus, Golgi complex and endoplasmic reticulum reappears. Next slide, you will see the diagram of this division. After the cytokinesis, cytokinesis means division of cytoplasm. So cytokinesis is the division of the cytoplasm of a cell after karyokinesis. Karyo means nucleus and cyto means cytoplasm. Karyokinesis divisions of chromosome into two daughter cells. In animal cells, the appearance of furrows in the plasma membrane deepens gradually and joins to divide cytoplasm into two parts. In the plant cells, wall formation starts at the center and grows outward to meet lateral walls. The formation of the cell wall starts with the formation of the cell plate. This is the diagram of number 1 is early prophase, then late prophase. Next one is this stage before metaphase start, and fourth one is metaphase. You can see all chromosomes are coming on the equatorial plate. जितने भी chromosomes हैं सब सेंटर में आ चुके हैं. After it एनाफेज. एनाफेज में आप लोग जानते हैं जो क्रोमोसोम्स हैं वो अलग अलग पोल्स पे खिंचना शुरू हो गए, तो उसमें शेप भी चेंज हो जाती है क्रोमोसोम्स की, वी शेप और जे शेप. आफ्टर दिस यू कैन सी टेलोफेज. ये जो न्यूक्लियर मेम्ब्रेन है ये साइड से इसमें कंस्ट्रक्शन बनना शुरू हुआ. सबसे पहले इसमें न्यूक्लियस डिवाइड हुआ, उसके बाद साइटोप्लाज्म, यानी कैरियोकाइनेसिस पहले होगा और साइटोकाइनेसिस बाद में होगा. टेलोफेज के बाद लास्ट वन इज टू डॉटर सेल्स आर प्रोड्यूस्ड. Significance of mitosis. Mitosis produces diploid daughter cells with identical genetic compositions. It helps in the repair of cells, especially in lining of gut and blood cells. Meristematic division in apical and lateral cambium results in the continuous growth of plants. Points to remember. The M phase starts with the nuclear division corresponding to the separation of daughter chromosomes termed karyokinesis. M phase ends with the division of cytoplasm known as cytokinesis. Next one is meiosis. In the meiosis, all stages are same, but only first stage, prophase 1, is the longest phase. So you have to remember, again I am repeating, prophase 1 is the longest phase of meiosis 1. The cell division that reduces the chromosome number in half and results in the production of haploid daughter cells is called meiosis. It helps in the production of haploid phase in the life cycle of sexually reproducing organisms. It involves the following events: two sequential cycle of nuclear and cell division called meiosis 1 and meiosis 2, but a single cycle of DNA replication. ये केवल एक ही बार होगा, डीएनए रेप्लिकेशन. It involves the pairing of homologous chromosomes and the recombination of them. 4 haploid cells are formed at the end of meiosis 2. Meiosis 1 and meiosis 2, same prophase 1, metaphase 1, anaphase 1, telophase 1. Like this, in meiosis 2, prophase 2, metaphase 2, anaphase 2 and telophase 2. Meiosis 1. In meiosis 1, metaphase, anaphase and telophase stages are the same as that of mitosis. Prophase 1 has 5 different stages. Number 1 leptotene, second is zygotene, third one is pachytene, diplotene and dikinesis. Out of these 5 stages, pachytene is very important because crossing over takes place in the pachytene stage. And in the second number is zygotene, synapses occur. It means 2 chromosomes paired. Prophase 1. In the leptotene, the chromosome become distinct and visible under the microscope. Compaction of chromosome continues throughout the leptotene phase. During zygotene stage, chromosome starts pairing together synapses. The paired chromosomes are called homologous chromosomes. Third one is pachytene. Crossing over takes place between 2 non-sister chromatids of homologous chromosome occur for exchange of genetic material. Diplotene is recognized by the dissolution of synaptonymal complex and tendency to separation of bivalent accept at the site of crossing over. This form an X-like structure called chiasmata. Dikinesis is marked by terminalization of chiasmata. The nuclear membrane breaks and the nucleolus disappears. Metaphase 1. In this stage, the bivalent chromosome aligns at the equatorial plate and microtubules from the opposite poles of the spindle get attached to the pair of homologous chromosomes. Next is anaphase 1. In this stage, homologous chromosomes separate but sister chromatids remain attached at the centromere. Last one is telophase 1. In this stage, the nuclear membrane and nucleolus reappear and cytokinesis follows. This is termed the dyad of the cells. This is the structure. Prophase 1, metaphase 1, you can see all chromosomes are on the equatorial plate. Anaphase 1, chromosomes move other poles. Telophase 1, 2 cells are formed. Meiosis 2. In prophase 2, the nuclear membrane disappears and the chromosomes become compact. At the metaphase 2 stage, the chromosome align at the equator and microtubules attach with the kinetochores of sister chromatids. Anaphase 2 starts with the splitting of the centromere of each chromosome to move towards opposite poles, and meiosis ends with telophase 2, in which 2 groups of chromosomes get enclosed by a nuclear membrane, followed by cytokinesis to form a tetrad of cells. Tetrad means 4 haploid daughter cells. This is the structure of meiosis. And in the end, telophase 2, 4 cells are formed which are haploid in nature. Significance: meiosis forms the gametes that are essential for sexual reproduction. Crossing over introduces new recombination of traits, helps in the maintenance of chromosome number of sexually reproducing organisms, provide evidence of the basic relationship of organisms. Last one is, question arises, what is mitosis, what is meiosis, what is cytokinesis, what is karyokinesis, what is equational division, and what is the difference between cytoplasm, protoplasm and nucleoplasm. So this is the last slide. We will meet tomorrow again. Thank you.